Hi, my name is Obi Bichukwu. Um, I'm in Group B, and in our experiment, we tested the pH effect on a particular enzyme and a particular substrate. Enzymes are biological protein formations used in chemical reactions. By this, I mean enzymes are catalysts that alter chemical reactions. In this case, we can say the way in which they alter chemical reactions is by lowering the activation energy. Alright, in this manner, activation energy means the minimum rate of energy needed for a reactant to carry out a particular chemical reaction. In our experiments, the enzyme used was trypsin and the substrate used was azocasein. Trypsin is a digestive enzyme found in the human gut at a pH level of approximately 8 to 9 and a temperature of 37 degrees Celsius, which is the normal body temperature. While azocasein is a casein with sulfanilamide groups covalently bonded to peptide bonds. Now, why do we need enzymes in our body? We need enzymes in our body because enzymes help with our daily, day-to-day metabolic activities such as digestion and respiration. During our reaction, we noticed the enzyme trypsin helped catalyze the hydrolysis of peptide bonds by breaking them down into smaller molecules in proteins. By breaking them down into smaller molecules in proteins, in which amino acids are then liberated, which makes it quite easier for it to be absorbed into the bloodstream. Our question for the experiment was to find the best pH range for trypsin to act on azocasein, and by this we came up with an hypothesis that the best pH range for trypsin to act on azocasein would be pH 9, and this was because, once again, trypsin is found in the human gut at 37 degrees Celsius and approximately level of pH 8 to 9. First of all, with our material methods, we laid down in a rack 12 microcentrifuge tubes, labeling them with their various pH levels of pH 11 to pH 6 to 11, I mean. Then we added 450 microliters of 0.15 NaCl reaction buffer to the distinguished pH levels. And then after that, we added 500 microliters of azocasein into individual 12 microcentrifuge tubes. So, after this, to create our experimental trial, we added 50 microliters of trypsin into 6 of the microcentrifuge tubes out of 12, and for our negative control trials, we added 50 UL of water instead of trypsin into the remaining 6 microcentrifuge tubes. We did this to test the difference between an enzyme in a reaction and without an enzyme in a chemical reaction. So, immediately after this distribution of trypsin, which was done really quickly, we incubated the samples, both the negative control trials and the experimental control trials, into a 40 degrees water bath for 20 minutes. Immediately after the incubation, we added 500 microliters of TCA to all the samples. And after centrifugation, we pipetted out the supernatant of each sample into clean cuvettes. By doing this, we took necessary lab protocols by not touching the bottom of the cuvette or smudging the sides of the cuvette. Then we added distilled water into one clean cuvette and placed it into the spectrophotometer in order to blank the cuvette so it would read at an equal range. Then we added each supernatant into 12 cuvettes. Then we placed the cuvettes into the spectrophotometer at 440 nanometer. And after all this, we read the absorbance levels of each supranatant equivalent with their different pH levels, and we did this to calculate the average absorbance levels, and the t-test, which is comparing pH 9 to the remaining pHs. Now, as we can see in our graph, from pH 6 to pH 7, there's a really huge difference, which is the same with pH 10 and pH 11. But when we come to pH 8 and 9, we can find out that there's no significant difference, which also shows in the graph because these levels are on the same range. So we couldn't say either pH 8 or pH 9 which one of them is the best pH level for trypsin to act on isocaine. So conducting the t-test here, we can find out that all p values apart from pH 9 and pH 8 are less than 0.05, which allows us to be 95% confident that our experiment was statistically different. Our hypothesis predicting that pH 9 is the optimum value for pH for trypsin to act on isocaine was not supported because of the not statistically significant value of 0.787, which is the t-test when you compare pH 9 to pH 8. So this indicates that this was an overlap. So as I said before, we couldn't particularly say pH 9 was the optimum pH value for trypsin to act on isocaine. So we based on still approximately pH 8 to pH 9, either of them to be the best optimal value for trypsin to act on isocaine. Thank you.